Hi, everyone. So I'm Jess, staff machine learning engineer at Recursion. I lead the ML infra team. This is Isla, who is joining me. We're on the same team together, and she's a senior engineer. So today, the purpose of this talk is to talk about what it takes from an infrastructure perspective, from a techniques, talent, culture, and practices to train and deploy large deep learning models. To kick us off, I'm going to hand it over to Isla, who's actually going to tell you a little bit about Recursion.
Thank you, Jess. So, Recursion. We are a clinical stage tech bio company that's leading the space by decoding biology to automate drug discovery. So, traditional drug discovery methods are inefficient and extremely expensive. 90% of drug candidates in clinical trials just fail. And if you happen to get a drug approved and be able to move it into patients, into actual treatments, it costs about $2 billion in total to get you there. Those costs are really high and really unmanageable. Recursion is looking to make this whole process more efficient by harnessing breakthrough technologies like the foundation model we're going to talk about to accelerate this process and try to break down the complexity of biology so that we can navigate to better treatments. Our goal as a company is to fail faster and fail earlier so that we spend less money and go faster. Currently, Recursion's time from drug discovery hit to validated lead is about 10 months on average, and that's about 3 times faster than the current industry average. How do we do this? How do we harness these breakthrough technologies? We built a thing that we call the Recursion OS. This allows us to collect high-quality data that collects a great deal of relationships between the different levels of biology and search them in an automated way. So we have an infrastructure layer of enabling hardware and software. We have a data universe that includes diverse and rich data sets, including the largest built-for-purpose biological and chemical data set in the industry. And then we also build maps that allow us to navigate the interconnectedness of biological data. So just a few proof points about the work we're doing. The size of our data set is over 25 petabytes. We have a 1.7 million small molecule library that's extremely large. We have 50 different human cell types. We generate 850 billion human-induced pluripotent stem cells in our labs. We've produced this many since 2022. We can run up to 2.2 million wet lab experiments per week. We can sequence 24,000 whole exomes. Exomes are protein encoding genes per week in our transcriptomics platform. We can leverage our top 125 supercomputer called BioHive 1 to do model training. And this is how we are harnessing breakthrough technologies to make drug discovery go faster. Oops. Hang on. I don't think our video is playing. Okay. We were going to show you a video of our wet lab with robotics that basically we have plates that have wells that are filled with cells that are treated with chemicals, chemical agents, and then stained and fed through high throughput microscopes. Unfortunately, it's not playing, so you'll just have to imagine this. The robots are very cool. So this is what I'd like you to imagine our maps look like. Imagine a world where a scientist can just go ahead and search through this highly relatable set of data in a really easy way to navigate the path for a disease from disease condition to healthy. So like Google Maps generated and organized a vast amount of data to find the best routes to navigate through traffic and around your cities, Recursion is building these maps to navigate through the complexity of biology and find a way to go from that disease state to healthy. The video would have shown you the technique that we call phenomics, where we analyze cells that have been perturbed with a chemical agent or a genomic agent, and we analyze the effects that those have on the cell's observable traits, on its structure, on its size, et cetera. The other layers of the map are starting to include things like data at the organism level, at the molecular level, and at the protein level, also genetics. All right, so that gives you some context and some background, but really, what did we actually build? Let's talk about that foundation model. Oh, and I'm happy to announce that the paper whose headline we're showing here was just accepted at NeurIPS as a workshop. So we're super excited about that. So what we actually built is a vision transformer that's been pre-trained with a masked autoencoder method. So a VIT and MAE model. What that means is we take input images, we divide them up into patches, we randomly mask out parts of the patches and remove them from input data. The model learns the raw pixels that make up the masked out portions, and then reconstructs the raw pixels for those masked out portions and returns to us a reconstructed image. So in this way, the model learns how to recognize image visual content. We can then use that model to learn new visual content in new images, produce an embedding vector that tells us the features of all those new images. We take that through inference, and we take that embedding vector and feed it then through the rest of the Recursion OS to start building out our maps. So this is a visual example of what that process looks like. We start with some target images. It's kind of hard to see the masks because they are rendered in black, but they're there. And then we get back some reconstructed images. Here's an example of building out a map. This is a whole genome arrayed CRISPR knockout map made up of data from human cells. What this means is we use CRISPR to knock out individual genes in cells. We put these cells through the automated high throughput screening platform, get images out. Then we do inference on the images, get out the embedding vector from the model, and then with some downstream processing, measure what the phenotype of the cell looks like because of the gene knockout. So we study the effect of the remaining genes expressions of the cell's genes and their interaction with the environment. This can have effects on observable traits of the cell, like size, form, structure. So that's represented with an embedding vector. We collect all the embedding vectors. We take similarity measurements between these embedding vectors. We visualize them as a heat map, And in the heat map, dark red means that the effects of the various gene knockouts are similar to each other, and dark blue basically means the opposite. All right, so I'm going to hand this back over to Jess to tell us how we built the foundation model.
So in this segment, I am going to talk about data, compute, and people. But as you can see leading up to this, Recursion is about a 10-year-old company. It took a really long time for us to generate all that data, figure out all the automation in the lab, install our supercomputer, also hire the team that can actually build these models. So how do we translate vision into reality? In 2019, Recursion put out this Kaggle competition, cell signal, how to disentangle biological signal from experimental noise in cellular images. This is, along with this Kaggle competition, we released RxRx1, which is, at the time, one of the biggest open data sets that's available if you're in this space. So RxRx1 had about 125,000 images and about 1,100 perturbations, classification labels. The winner of this competition, we took the model, we hired the person that actually won the competition, and we released our first deep learning model in 2022. It's a DenseNet. We chopped off the final classification head, and we have these embeddings that we feed through, again, our pipelines. So each image, we learn kind of the representation of what's happening at the subcellular level, so genes and compounds. In 2021, we held the data set at the same size, so we have a mild improvement in which we could compensate for these batch effects. Batch effects, if you're unaware of what that is, there are things that happen in the physical lab, so like there could be temperature, it could be humidity, there could be various factors that are not biological, but just contextual physically. But we used adaptive batch norms to actually gain better domain adaptation for our embeddings, and we could see this throughout our metrics. In 2022, we developed a whole series of internal data sets of varying sizes trying to scale up this model, but something weird happened. Our models started performing really well in certain benchmarks, but really poorly in others. And we're like, what's going on? This actually led to a really, really cool insight, which there's another paper that we published recently on proximity bias. And what proximity bias is that we found that the gene's location actually mattered as much as its function. And so when we're using CRISPR-Cas9 to make this gene editing, what we see is that even though we cut out this gene, we could see morphological changes for unrelated genes that are just close in proximity in the same chromosome arm. Back to our poor ML team, though. They felt like Sisyphus, like, what's going on? Like, how do we actually improve on our model? We're trying to increase the amount of data we're learning from, but this is not doing what we're expecting. So we ditched dense nets. We started looking to the stars. We started looking at variational autoencoders. We started looking at vision transformers in general. We tried a lot of different architectures before we actually landed on masked autoencoders. And so in 2023, we did find early success with autoencoders, and we started scaling up with even more data. The 53 and the 95 is actually the number of wells we have. And I'll talk a little bit about that later. But this year, we also released RxRx3, which is, again, the largest open data set that is publicly available. You can go on rxrx.ai right now and download these 2.2 million images. We are a big proponent of contributing back to the research community, and so that's what we do. I have a pop quiz for everyone. You have a supercomputer. It can ingest data over the internet at about 10 gigabits per second. How long does it take you to transfer 4 petabytes of data? Conversion here. Anybody want to take a guess in the audience? Okay, I'll give you a second here. All right, so if you guess 35 or 37 days, you're correct. But you don't want to be that jerk that also cripples your supercomputer because it can't connect out to all the other services that is required for deep learning. For example, if you're tracking your experiments or you need just to report metrics back to your server. So if you're flying at about 80% utilization, that's 46 days, that's half of your quarter, assuming you're working on the weekends. Not great. What can you do to make this faster? So most of our data actually lives in the cloud. GCP is our main service provider. In collaboration with them, you can actually dig up some dirt, put some fiber cable down, and also share this co-location facility with them and get direct access to their services, private addresses. And if you do this, you can 4x your transfer speeds between your cloud provider and yourself. So cutting down that 46 days to about 11 and a half days. Second pop quiz. You have 320 A100s available. Our supercomputer is consisting of 40 nodes of DGX A100s. NVIDIA is great. Highly recommend working with them. In this case, each A100 has about 2 terabytes each, so you have 640 terabytes you need to chew through. So that also means you need fast enough file systems to actually, for your CPU to actually run ahead and kind of load it up and, tee off your GPUs. But, that's in total, if you have 4 racks of NVMe drives, you're only looking at about 320 gigabits per second. That's not that fast relative to what your GPUs are demanding of you. Another way you can also do this is to use distributed training. So there are various strategies available. We found, at least for like the low billions of parameter of models, you can get by pretty well with just data parallel. That's one of the simplest strategies for distributed training. Pretty easy to get set up. And if you don't know what data distributed is that you are broadcasting all of your parameters to each of those GPUs. Each of those GPUs computes the gradients. You have to reduce, so there's some communication bottleneck there where you actually have to, optimize, and then you send out those parameters back to those GPUs, and they upgrade or update those weights. So going back to those plates that we have, another way you can also speed up data loading overall is just storing your data in an efficient format. So this is an example of a plate we have in the lab. Each of these plates has about 1,500 images. Each of those images is actually, each of those wells are actually 6 images, one of each channel. Certain channels will pick up on nuclei, some channels pick up on mitochondria, etc. But from this, you can actually smash your 6 images into a multi-dimensional array. We're using the PyData stack here, so these things called ZARs. They're chunked, they're indexed, which just means that when you're loading random crops of images during the training loop, you can actually seek to exactly where you need to load your data. And here you see a graph of our GPUs very happily humming along close to 100%. Those spikes, I suspect, is actually when we're checkpointing saving our checkpoints. I'm going to move over a little bit to compute, but I'm also going to step it back a little and talk a little bit about Ehrum's law. So why are we doing this anyways? Our data that we generate in-house is bounded to atoms. You actually have to buy cell lines, you actually have to buy chemical compounds, and you actually have to do physical experiments. That's a lot more expensive than the bits and bytes that are running through our LLMs. As Ala mentioned earlier, a drug costs billions of dollars to develop. Models are still in the millions of dollars. So as Ehrum's law states that drug discovery is getting slower and more expensive, we're trying to combat this with Moore's law, where computing is cheaper and less expensive over time. And so as part of this process, we also train a bunch of models, hundreds of models, trying to search for the right hyperparameters, the right data set, just the right recipe. Here on the left there, you can see that we have public data, small models, and pretty good recall, but just to the right of that, we try to scale up the data but holding the same model size, and you're spending more flops, but you're not really getting that much more performance. However, it's when you scale up both your data, the model size, and throwing way more compute at it, you get the better lesson once again, which is just more data, more compute, more performance. So before we had our supercomputer, we actually had a bunch of 1080s. I don't know if you all remember. These were like gaming consumer grade GPUs. They actually worked great. We had hundreds of them. At the time, we were using PyTorch Lightning. This is around 2020, and it was a pretty immature project at the time. It didn't do exactly what we wanted to, but we had a very small ML team. They didn't have any engineering, so it was a relatively small team. We adopted Determined AI because it was an all-in-one deep learning platform, and they had great support. They had experiment tracking, resource allocation, checkpointing, fault tolerance, centralized logging, all that good stuff. We also started dabbling around with some V100s, but ultimately, by the time we bought our supercomputer, they were mostly A100s. So in 2021, when we installed our supercomputer, we were running Lustre as a file system. This was pretty bare bones. We were still using Determined, and also we used Prometheus for making sure that our supercomputer is acting healthily. We actually did a fairly large rebuild last year where we adopted infrastructure as code. We were using Bright Computing to provision our nodes. We have some Ansible playbooks. We have a lot more operations with and monitoring with Prometheus. We also got rid of Lustre. I don't know if y'all have deployed that many file systems, but for the reliability, scalability, and also the performance that we needed from our file system, we switched over to Spectrum Scale. And lastly, for our scheduling needs, we switched over to Slurm. So if y'all aren't familiar with Slurm, this is probably running in most of the supercomputers in the world. But it also plays much more nicely with different workloads that's not just deep learning and determined. So we do other things on our supercomputer as well, but training models is definitely a big part. Moving up the stack, though, we also rewrote a lot of our training workflows. So before we used Determined AI as our training harness, we replaced that altogether with PyTorch Lightning, partly because newcomers that were coming into Recursion, as we were hiring out our team, we noticed that, well, Determined's not very widely adopted, but also PyTorch Lightning was getting a lot of momentum. So we revisited this decision again, and we actually found out it was a great fit for us and also simplified a lot of our code. One of the really cool things about PyTorch Lightning as well is that you get a lot of optimizations out of the box for free. So for example, if I'm looking at distributed training, with Determined AI, they were based on Horovod. We got rid of that altogether, simplifying our overall dependencies, less dependency Python hell. And on top of that, we moved towards just using Torch Elastic, Torch Run, Torch Distributed, that whole ecosystem. Another thing we did in terms of being able to make sure we can reproduce our results was to adopt Hydra. So Hydra is another library from Facebook where you can compose your experiment configuration, and we went from thousands of lines of glue code for our submission scripts to much less. We got rid of that submission script altogether. Overall, though, highly recommend it. Last thing we did most recently was actually upgrade to the latest version of PyTorch. So if you're not already on PyTorch 2, highly recommend it. Again, if you're doing any kind of transformers-based training, you'll get flash attention just out of the box. And lastly, we were also noticing on the inference side of things, we were blowing past all of our runtimes and all the service levels that we had set out with our old model. And so working closely with NVIDIA, we've also been optimizing our workloads with TensorRT. So this is when we used to go from TorchScript, we can compile that and convert that down to TensorRT, which is more optimized for NVIDIA's hardware. We also added new GPU types. We run a lot of our inference on T4s. It's cheap. We have a lot of inferencing needs. But we also started mixing L4s in there. We found that we could get 2x the input with less money for the buck, or better bang for your buck. I'm going to pass it over to Isla to close us off.
Okay, so you can't do any of this work if you don't have the right team. This is the team that built the foundation model. We came together with a lot of diverse skill sets. We have ML researchers, we have engineers with machine learning, data, and software engineering backgrounds. We came together in work streams so that we could break down silos between all these different skill sets and functions. And that allowed us to iterate pretty fast because communication is a big bottleneck in a large and complicated project. One of the other benefits of having work streams is we could kind of self-organize into so that we could deliver our goals fairly quickly. We minimized handoffs between all the different parts of the project so that we could deliver on our goals quickly. We also relied on teams in other parts of Recursion. The high-performance computing team that manages the BioHive supercomputer was absolutely key to our efforts to train this model effectively. Then once we actually have the model, we collaborated with the teams that look after the wet lab experiments, the high-throughput screening teams. We needed to integrate this model into the workflows that process new experiment images and produce embeddings. And then we also worked with the team that builds out the maps. We had to backfill a bunch of experiments that had been previously executed in order to build the maps. So that concludes our talk. Thank you for your time and attention. I think we may have time for a little bit of Q&A. We can take one question.
Hello. Hey. For people that are starting out in computer vision, what do you think is the most important thing to help you improve your model? Was it the data? Was it the improving the data, improving the model? What do you think led to the highest performance increase?
So yeah, definitely more compute, more data, but not just more data, but better data as well. So we also did various optimizations of like sanitizing some of our data, deduplicating it. We have a whole data review process in-house also to make sure that like our images are good coming out of the lab. So when you're taking 1,000 pictures at the same time, you might not get what you expect.
Awesome. Thank you, guys. Why don't we give them a round of applause?